You know that smell of fresh coffee that just hits you in the morning? Or the scent of rain on dry pavement? Smell is, well, it's one of our most ancient and powerful senses. But have you ever stopped to think about how it actually works? How does a tiny molecule floating in the air turn into a feeling or even a memory inside our brain? Well, today, we're going to decode that exact puzzle.
So, it all kicks off with an odor molecule, right? Something released from, say, this flower. It makes its way into our nose, or if you're an insect, your antenna. But how does the brain know the difference between a rose and, I don't know, gasoline? How does a physical thing get translated into a neural signal? That's the question that's kept neurobiologists like me fascinated for decades.
To figure this out, scientists uncovered this remarkably elegant principle. It's a fundamental rule that seems to explain how our entire sense of smell is built, right from the ground up.
Just think about it for a second. We could tell the difference between thousands, maybe even 1,000,000,000,000 different smells. I mean, that's a staggering number. So the brain needs an incredibly specific and organized system to keep all those signals from just turning into noise. So how on earth does it manage that?
This right here is the key. It's called the one neuron, one receptor rule. And the idea is actually pretty simple, but it's incredibly powerful. Each individual sensory cell in your nose, we call them olfactory sensory neurons or OSNs, is a total specialist. It only makes one single type of receptor, which is designed to catch a very specific family of odor molecules. And the absolute poster child for this rule is the mouse. Now, what's so great about this slide is how it visualizes this idea. Just imagine each of these colors represents a different kind of odor receptor. You see how in the mouse, each neuron, each one of those little circles, is just one solid color? One neuron, one receptor type. There's no mixing, no confusion. It's just a beautifully clean and strict system. And that organization doesn't stop there. All the neurons that are the same color, meaning they all have the same receptor, they all send their signals to the exact same spot in the brain's olfactory bulb. These spots are called glomeruli. So all the blue neurons report to the blue spot, the red to the red spot, and so on. This creates this incredibly precise map where each location stands for a specific odor feature. It's a brilliant way to sort all that information before it even gets processed.
Okay, so this elegant rule seems like a perfect solution, right? And when scientists turned to another powerhouse model for neurobiology, the humble fruit fly, they pretty much expected to see the exact same playbook.
And here it is, Drosophila melangaster. This tiny insect has been an absolute giant in scientific discovery for more than 100 years, helping us unlock secrets of genetics, the brain, you name it. Its nervous system is way simpler than ours, but it has to solve all the same problems, like using smell to find food and stay out of trouble. And, you know, when researchers first started mapping out the fly's sense of smell, it looked like they had found the same rule all over again. Neurons in the antenna pick up smells, send their signals to glomeruli in a part of the brain called the antennal lobe. It really seemed like evolution had just found the one best way to do this, but then they looked a little closer.
And this is where our story takes a really surprising turn. It turns out the fruit fly isn't such a strict rule follower after all. It has a more flexible, and in some ways, a much more complex strategy.
So the whole thing boils down to this contrast right here. The mouse system is rigid, one receptor type per neuron, end of story. The fruit fly, our little rebel, well, it usually follows the rule, but it has some really important exceptions.
And this just illustrates the difference perfectly. On the left, there's the mouse again. Each neuron is a single, pure color. But now look at the fly on the right. Sure, a lot of the neurons are a single color, but you see those others? The ones that are speckled with multiple colors? That means a single olfactory neuron in the fly can actually express more than one type of odorant receptor.
Now, this isn't just my interpretation. This is a direct finding from the research. As one source puts it, in flies, there are some exceptions to this odor coding rule. An olfactory sensory neuron will express two different odorant receptors. Suddenly, the rule wasn't quite as universal as everyone thought.
So what we're seeing here is a phenomenon called co-expression. As this diagram shows, instead of having one neuron for the red receptor and a different one for the blue, the fly can just pack both the red and blue receptors into a single neuron. What's so fascinating, though, is that even these rule-breaking neurons still connect to a single glomerulus. So the brain's overall organization, that map, it stays intact. It's just a different way to build the detector in the first place.
And this isn't just some rare one-in-a-million thing. This co-expression strategy happens in about 10 to 15% of the fly's olfactory neurons. That's significant. It's an intentional part of its sensory toolkit.
So now we have two animals, a mouse and a fly, with two completely different strategies at the cellular level. And this brings up a much bigger question. Why? What's the evolutionary logic here?
I mean, why have a super strict system in one animal and this flexible mix and match system in another if the end goal, smelling the world, is the same? Well, for a mouse, maybe that strict map is vital for picking up on subtle social cues. But for a fly, maybe having a single neuron that can detect a combination of chemicals from a ripe piece of fruit is just a much more efficient shortcut.
And this is the really beautiful answer. Despite all those differences we just talked about in the individual neurons, the overall wiring logic of the olfactory system in mammals and insects is just stunningly parallel. This is a classic textbook case of convergent evolution, where totally different evolutionary paths lead to a remarkably similar solution.
Look, whether you're a mouse or a fly, the process follows the same three basic steps. First, sensory neurons out on the periphery detect the odor. Second, their axons all converge on that primary sorting station, the olfactory bulb or the antennal lobe. And third, that sorted signal gets sent on to higher brain centers for interpretation. The fundamental logic is the same. It's been conserved.
So what does all this tell us? Well, to me, it's a story about the pure ingenuity of evolution, finding more than one way to solve an incredibly complex problem. Ultimately, both the mouse's strict, by-the-book system and the fly's more flexible one achieve the exact same goal. They transform the chemical world into a coherent neural map. This allows the animal to make sense of its environment, one single molecule at a time.
Which leaves us with a really fascinating final thought. Is there ever really one best way to build something as complex as a brain? Or is the real genius of evolution found not in a single perfect solution, but in its incredible flexibility to find different but equally effective paths to the same destination?